Hi, welcome to this video from Doc Hill Study. I want to explain what's going on here. So I'm a teacher in Scotland, but I'm also a registered teacher in Queensland, Australia. And I've been over here since 2019, so I've missed the start of external assessment in Australia. So what I'm going to do some past papers from the Queensland Government, QCAA, the Queensland Curriculum and Assessment Authority. And this provides a bit of a double purpose. The first thing it'll do is it'll give me a little bit of a keep up with the new assessment style in Queensland, and give me an opportunity to get my head around what's going on there. And also, I think that it could be very useful for those people studying for exams in 2025. The exams are coming up this November, I would guess, and with any luck, this will be useful to you in your revision. So I'll put this up basically as a revision video to help you with your learning. It says here, which term describes a group of organisms consisting of a common ancestor and all its lineal descendants? And so we look at this, well, population and community are both ecological terms, so we can immediately rule these out and this out. Subgroup is a bit of a vague term, and what we're looking at in our biology and our systematics learning is clade. So I'm going to put clade as the answer. This is, that's what it says, a group of organisms consisting of a common ancestor and all its lineal descendants. Question 2. The polymerase chain reaction is a technique used in DNA profiling to, and we've got several choices here, cut DNA at specific sites. Well, you need an enzyme to do that, not a reaction. To create more copies of DNA, that's what we do with polymerase chain reaction, PCR. It makes more copies of the DNA, doesn't determine the order of nucleotides, and it doesn't separate DNA fragments based on size. So we're going to create more copies of DNA. Question 3. What is the key difference between spermatogenesis and oogenesis? So this is a tricky one now. Spermatogenesis produces haploid cells, whereas oogenesis produces diploid cells. Well, the ova that's released from the ovary does have 2 copies of DNA in it, but it's not technically diploid. Spermatogenesis produces 4 functional gametes. Well, that's true, whereas oogenesis produces 1. That's also true. See if there's any better answers here. Spermatogenesis occurs throughout life. That's true, whereas oogenesis only occurs after puberty. Oogenesis begins before birth, in fact, and finishes in the fertile period of a woman's life between puberty and menopause. So I'm not going to go for that. And spermatogenesis, of course, doesn't occur throughout life either, because it doesn't occur before puberty, and it does continue for the rest of life after puberty in men. And spermatogenesis begins with haploid cells. No, it doesn't. Whereas oogenesis begins with diploid cells. That's true, that's false. Okay, so we're going to go for spermatogenesis produces 4 functional gametes, whereas oogenesis produces 1. The idea is that oogenesis is made up, the ova is made up really of 4 cells, which keep getting squashed together so that the cytoplasm of the cells grows and produces all the nutrients required for the egg. Okay, question 4. The table shows data on 4 plant species, which is most likely to be a pioneer species. Well, what's a pioneer species? Can we remember? Well, a pioneer species is one of the species that grows up first after some disaster, like a volcano erupting and killing everything in the area, or perhaps a tree falling in a forest, creating a light zone gap in the canopy. And the plants that get going there are what we call the pioneer species. Okay, so what they need to do is they need to get going as quickly as possible. They need to grow as fast as possible. And ideally, they should have nitrogen-fixing bacteria in their system, so that if the area has been denuded of nutrients, they can get their nitrogen from the air via the bacteria. So I want a rapid germination rate, and I want the presence of nitrogen-fixing bacteria for ideal pioneer species. And the ideal pioneer species is going to be rapid and present, so I'm going to go for C. Question 4 is C. Okay. Right, question 5. A limitation of the biological species concept is that it does not account for species that, okay, reproduce asexually. Well, that's the classic one, because the idea, the biological species concept, remember, is if two individuals can interbreed and produce fertile offspring, viable offspring, then they are the same species. Now, if you reproduce asexually, you can't do that test, so it doesn't really account for that. Reproductively isolated, no. Are morphologically indistinguishable? No, that means they look the same, but it's not a problem with the limit. It's not a limitation of the concept. Have different number of chromosomes. That would make them different species. So that doesn't work because they're already different species, right? Okay, so never say never in biology, but I don't think that's going to work. Anyway, we're going to go for reproduce asexually, because this is not covered by the basic species concept. There are other ways of defining species, though, of course. What can introduce new alleles into a population? So let's have a look at the options. Point mutations, non-disjunction, random fertilization, and independent assortment. Well, independent assortment creates variation, but it doesn't create new alleles because it's not breaking the DNA. It's not splitting the DNA. Random fertilization, I mean, we haven't got crossing over here. It can sometimes bring new alleles. So this is one of our elements of variation. Independent assortment is entire chromosomes being sorted out. Random fertilization, that doesn't do anything for alleles. It doesn't change the DNA. Non-disjunction, this is getting the chromosomes mixed up, but it again doesn't break up chromosomes. And point mutations, this will change the DNA on a gene which produces a new allele. Could produce a new allele, doesn't necessarily have to. A point mutation can sometimes be silent. So we're going to put that one. That can do it. Doesn't necessarily do it, but it can do it. That's the only one that will. All right, let's move on to question 7. Oh, here's a big one. Let's have a look at this. Right, tolerance ranges for species X and a Holdridge life zone classification chart are shown. Here's the tolerance. Let's have a quick look at the tolerance of the species X. X marks seems to have an optimum survival temperature of 10 degrees Celsius and an optimum survival growth rate. These are growth rates for plants, not survival, but growth rates. Optimum growth rate at 10 degrees Celsius and optimum rainfall zone of 400 millimeters of rain per year. Okay, so what we have to do is take that. We've got 400 here for rain, and we'll write it down here so that we can see it. Temperature 10 degrees Celsius. Now what we have to do is interpret this thing. Now this chart here has a couple of axes on it going in triangles. The axis along the bottom is really just the descriptors. So let's see. We have annual precipitation, potential evapotranspiration ratio. Well, we don't really have anything up here that's helping that needs this, so we're just going to look at annual precipitation. And we want to be in the 400 zone, so it's going to be this one here, in between 250 and 500. It's increasing downwards here. And we want to go down here. So we've got desert, wet tundra, moist forest, or steppe are the 4 possibilities for this. And those are the 4 possibilities they've given us in the options, I think, pretty much. Desert, wet tundra, moist forest, and steppe, correct. Okay. So which one do we choose? Well, we want the one that's going to most likely have a 10-degree temperature in it. So desert is 1.5 degrees Celsius to wet tundra, desert to wet tundra, to moist forest is 3 to 6, and steppe is 6 to 12. So I think we're going to go for steppe, which is B. Right, we're going to take that one. All right, in the Linnaean system of classification, 2 species of the same class will also belong to the same what? Okay, so if you remember, it's kingdom, phylum, class, and then order. So if you're in the same class, you must also be in the same phylum, but you're not necessarily in the same order, right? Got that? And then below that we've got family and genus and species. Okay, so we want to pick phylum or kingdom. Kingdom's not an option that we're given, so they have to be of the same phylum. And the answer is D, question 8. All right. So I'm talking through all my thinking on these questions to try and help you out with your revision. The idea is that each question is a little kind of mini lesson, almost, and a little mini lesson to me. And it reminds me what comes up in this is on the curriculum for these assessments. For example, the detail of the spermatogenesis and oogenesis is new to me. That we had in question, what was it, question 2? Anyway, the graph shows how populations of two species living in the same area change over time. All right, so there's the population, there's time, species Y, boom, species Z, ooh, it's kind of not going well, species Z. They were going okay for a while, and then suddenly we reached some kind of critical population here, and this species kept going and the species died out. Now, what's going on in this graph? Well, the most likely thing is that they're using up all the, at this point, the resources in the environment become scarce, and the two species become competitors for those resources. Whatever the resource happens to be, it's not necessary to know what it is. But at this point, below this point, the two species are going okay because the resources are plentiful, and they haven't reached the limit of resources. And then, as soon as they hit this point, they're in competition for the resources, and species Y is outcompeting species Z for those resources, and therefore species Z eventually doesn't reach any form of critical population size and will eventually peter out and become extinct in the area. Okay, so what species interaction is indicated? Well, we've talked about this, and it's competition. Let's have a look. Predation, where species Y preyed upon species Z. This is not the case, because what you tend to see is a couple of lines that peak and trough alternately from species Y and species Z for predation. Predators obviously don't want to wipe out their prey, otherwise they'll have nothing to eat. So the predator-prey relationship is not a relationship that generally leads to the extinction of one of the species. That's just a general thing about predator-prey relationships. Now, it can happen when new predators are introduced into an environment where they've never been before, but under normal circumstances, predator-prey relationships are fairly stable, and the populations increase and decrease alternately depending on the populations of the predator or population of the prey, right? So, right, predation, where species Z preyed, that's obviously not correct. Competition. Now, we're happy with competition here. We've already talked about it. They're fine up to here. When the resources start becoming in short supply and they're competing for the resources, and the species Y outcompetes species Z. So species Y outcompeted species Z. We're going to pick C. Competition. So they're just given this competition and predation either way around, and just make sure you don't pick the wrong one of these two. So that's a question of just making sure your literacy is good enough to understand the sentence here. All right, be careful with those. Question 10. A key function of histones is, right? Well, the histone is really a structural protein that allows DNA to wind, it packages up DNA. So let's see. Adds nucleotides to a growing DNA strand. Well, it doesn't do that. What does that? That's DNA polymerase does that. Binds to specific sections of DNA to initiate replication. No. I'll have to try and remember in my head what's doing that one. Allow DNA to tightly condense so it fits in the nucleus. That sounds good. Joins amino acids to form a polypeptide chain during translation. No, that's the ribosome does that. It allows DNA to tightly condense, and that's your answer for number 10, C. Let's have a look at question 11. Which mode of inheritance leads to continuous variation in the phenotypes of a population due to the cumulative effect of multiple genes? Right. So the cumulative effect of multiple genes here is the key. This is polygenesis, or many genes having an effect on the phenotype, and the continuous variation comes from, well, it's not co-dominance, because that tends to be just a couple of individual genes. Incomplete dominance, again, a couple of individual genes working together. Multiple allele inheritance. Don't think so. I'm going to go for polygenic inheritance. Polygenic, basically multiple genes, polygenic. Multiple allele is not multiple genes, so that's multiple alleles of one gene, and this is multiple genes giving multiple phenotypes that kind of mix together and give you a continuous variation. So I'm going to go for B for number 11. Number 12, oh, here we go, nitrogen cycle, we love it. Okay, the diagram shows key processes in the nitrogen cycle. What have we got? Nitrogen, this is a nice little diagram, I like it, I might use it in my own teaching. Looks good. Lightning, good. Okay, so we have nitrogen fixing bacteria, and they produce something. Then the nitrifying bacteria produce another thing, and more nitrifying bacteria produce a third thing, and the denitrifying bacteria produce nitrogen, and the third thing is used by plants. So what we really want to do here, and the decomposers produce this stuff as well. So what we want to do is remember what it is the plants use. This is nitrates, has to be nitrates. The decomposers and decomposing animals and plants, they produce ammonia. And in here is a step that goes in the process from going from ammonia through one type of bacteria to nitrite compounds, and then through another kind of nitrifying bacteria to nitrate compounds. So let's see. One should be ammonia, nitrite, nitrate. That's the order of things. So we're going to, but just, oh, they've given us a formula here, a chemical formula for each one. So maybe we have to check those are correct as well, do we? We're going to start with ammonium, right? Well, there we are, ammonium. All three of those are wrong then, because one is ammonium. So that's nice and easy. It's going to be D, right? Ammonium, nitrites, nitrates. Good. So question 12 is D. Question 13. The role in space that an organism fills in an ecosystem is referred to as its niche. That's classic question, and that's the classic definition of niche. Ecoregion, no. Environment, no. Microhabitat, no. All right, so compared to K strategists, species classified as R strategists tend to have, okay, well, this is the fire and forget type of thing versus care for your young. And I always think that the best way to remember this is that care for your young, just remember it's spelled with a K. So K strategists put an awful lot of effort into caring for their young to give themselves a good chance, give the young a good chance of survival. So what they do is they invest in fewer offspring, but put more care into them. Whereas K for care. Species classified as R strategists tend to fire and forget. Fire and forget is there's two R's in fire and forget, so there you go. But there's also an R in care, so it doesn't really work. Anyway, species classed as R strategists, what they do is they just produce lots and lots of offspring, put very little care into them, put all their energy into producing huge numbers of offspring, and as a result, the chances are some of them are going to survive. So the answer is A. Question 14, the answer is A. All right. Which graph best illustrates exponential growth? Okay, so we're looking for something that starts off slowly and increases and then keeps going. So this is fading out rather than growing. This gets wobbly. We want it so start slowly, gets going, and then levels off. That's not exponential because it's not keeping going. This one here is start slowly, increases, and then basically goes vertical, and that is exponential growth. There. Question 15 is D. Question 16. Oh, we've got a food web. All right. Diagram shows feeding relationships for an ecosystem where tiger sharks are keystone species. Well, I love Australian ecosystems. They're such exciting animals. We've got in Scotland they talk about things like rabbits. Okay, so let's see. We've got the tiger shark. We've got the spotted wobbegong, one of our favourites. The trout, giant triton, big shell that is. Starfish, butterfly fish, jellyfish, turtle, octopus, crab, coral, seagrass, and plankton. So these are our producers down here. And everything up here is our consumers. Coral are a bit of a mixture of a producer and a and a consumer, because they have algae in their cells which produce food for the coral, and then the coral. Yeah, so anyway, let's go here. What species is most likely to experience an increase in predation if tiger sharks are removed? Okay, well, if you take away the tiger shark, who's going to get more? Who's going to be predated on more, right? So the tiger shark, if present in this case, we need to think about this. The tiger shark, if present, is eating the turtles, it's eating the trout, and it's eating the butterfly fish, right? So if the tiger shark goes missing, then these three populations are going to increase. The turtle, the trout, and the butterfly fish are going to increase in population. And that means their predation on their prey will increase. So the predation on the jellyfish will increase. The predation on the butterfly fish might increase. That's a bit complicated because the butterfly fish are also going to be increasing in population. So that's a bit. But the predation on the coral might well increase because of the butterfly fish. Although the trout population probably is going to increase and start eating the butterfly fish. So let's see what choices we've been given. Trout. No, its population is going to increase. And it's now the top predator because the tiger shark is gone. Starfish isn't involved in that feeding relationship we're talking about with the tiger shark. Jellyfish, excellent. The turtle is going to increase in population because of the absence of the tiger shark. Therefore, the jellyfish is going to see an increase in predation because there'll be more turtles, right? And the spotted will be gone, again, a top predator, not interested. So we're going to go for jellyfish. Answer is C. Question 16 is C. Question 17. Which genetic tool is not required to make recombinant DNA? Okay, which genetic tool is not required to make recombinant DNA? Okay. So we need to ligate the DNA into the plasmid vector using DNA ligase. That's plasmid vector. Take your DNA, put it into the into the vector using DNA ligase. Restriction enzymes. Yes, transcription factors not needed. They're a nuclear protein, can interact with the nucleus and cause gene transcription. So they're not needed. So question 17 is D. These three are required to make recombinant DNA. 3 to go. Question 18. Look at this for a diagram. Karyotypes for 2 human gametes are shown. Here we go. Look at this. We've got a gamete here. Doom. Yeah. 2 X chromosomes. Excellent. Okay, so this gamete here, for some reason, has 2 X chromosomes. That's the unusual thing about this. Over here, this gamete is a perfectly normal female gamete, 1 X chromosome. If the 2 gametes produce a zygote, the resulting embryo would be female with trisomy, right? So it's going to have 3 X chromosomes in its cells. And the effects of that is something I'm going to have to look up. I don't know how much detail the curriculum goes into that. I guess we're going to find as we go through the various papers. But I'm going to go for question 18 is B. I'm going to go for that. All right. Coming at this a little cold. Actually, I haven't looked at the curriculum documents really to see what's on the curriculum for these tests. I just having to look at the paper to see. Which diversification pattern involves the independent development of similar structures in 2 species with no recent common ancestor? All right. So, what diversification pattern involves the independent development of similar structures in 2 species with no recent common ancestor? What would that be? Let's think of an example of that. Well, one example is the classic one is the vertebrate eye versus the cephalopod eye. Very, very similar structures. But they did not have a common ancestor that produced that structure. I don't know the word how recent comes into this anyway. Is that parallel evolution or is it convergent evolution? These are the 2 you're going to mix up. Okay. No recent common ancestor. Well, certainly octopus and humans. I'm going to call that convergent evolution. Okay, question 19. Now, you might pick parallel evolution here. Co-evolution is when something like a flower and an insect evolve together. Parallel evolution, I'd have to check. Divergent evolution is when structures differ very greatly. Convergent evolution is the answer to this. Okay, and question 20. Let's see. The graph shows the number of nucleotide differences observed in the cytochrome C genes of 10 species and their time since divergence. Okay, so cytochrome C doesn't really matter what it is. I think it's one of the mitochondrial genes, isn't it? And 10 species, so they're going to be fairly highly conserved anyway. It's a gene. And the 10 species time since their divergence. So these two, let's have a look at the graph. Nucleotide differences up here, time in millions of years. So these two species, each one of these spots is a species, right? So have a look at this. The cytochrome C genes from two new species were compared and found to have 60 nucleotide differences. So let's go to our 60 nucleotide differences. Make sure you read the right angle because these axes are almost the same scales. So you might just get them mixed up, which I just did there a second ago, but I've cut that bit of the video. So you're going up to 60 nucleotide differences and you're trying to figure out how long ago that might have been. So I'm going to go to about there, probably, maybe there. And that's going to make somewhere between 70 and 80 million years ago. What have they got here? 70 and 80 million years ago, and go for C. You could have mistakenly gone this way and gone for 48, which they put that answer in so that you can get trapped on that one. Watch out for that in the test. Make sure you're on the right axis. Very easy mistake to make. Okay, hope you enjoyed that video on the QCAA biology test. This was, which year was this? That's a good question. 2024. Okay, see you next time. Bye for now.